Hello, I'm Richard Young, your host with Facundo Batista and Lena Fan for MIT's course on COVID-19, SARS-CoV-2, and the pandemic. Despite the excitement about evidence for new and efficacious COVID vaccines, effective therapies are urgently needed. There are over 500,000 new cases of COVID-19 being reported each day worldwide. We heard last week about the development of small molecule drugs in this space. And this week, I'm delighted to say we'll hear about the development of biological therapeutics by one of the world's leaders in this area, Dr. Herbert Skip Virgin. Dr. Virgin's the Executive Vice President of Research and Chief Scientific Officer at Vir Biotechnology. At Vir, Dr. Virgin directs a team of scientists focused on immune approaches to prevention and treatment of severe infectious diseases. Dr. Virgin received his bachelor's degree, his MD, and PhD degrees from Harvard University, and then trained in internal medicine at Brigham and Women's Hospital in Boston and infectious diseases at Barnes Hospital in St. Louis. He served as professor and chair of the Department of Pathology and Immunology at Washington University of Medicine in St. Louis. While there, his lab used genetic, structural, computational, and sequencing methods to define mechanisms of viral pathogenesis and immunity. Dr. Virgin's a member of the American Society of Clinical Investigation, the Association of American Physicians, the American Academy of Microbiology, and the National Academy of Sciences. Skip, thank you for talking to us today about therapeutics development for infectious diseases.
Howdy, everybody. It is an honor and a privilege to speak at a class at MIT. I didn't actually realize that you let people trained in that other institution across town actually speak at MIT. So I'm profoundly honored and looking forward to it. So I'm just going to share my screen here and ask y'all, can you see the first slide here? So it's harnessing immunity to prevent, et cetera.
Looks good.
Okay, great. So thanks very much for the opportunity. And today I'm going to talk about therapeutics and prophylactics that are biological in nature. This slide has my title and some of disclosures of other companies than Vir, which I'm associated with. So all talks from companies have a legal disclaimer. This you don't need to memorize. It says that the things that I'm saying are at risk and may or may not turn out to be true or correct, as is true of all science and many things in medicine. So the goals that I'd love to accomplish today are to help folks understand the passive transfer of antibody as a therapeutic modality. Passive transfer means the administration of an antibody to someone who's not immune. The antibody functions that are relevant to that use of antibodies, some molecular immunology, if you will, some new things that have very recently been published that we've participated in about how to engineer antibodies for enhanced function, and then something that I find a bit more sobering in the setting of the pandemic that you've been learning about, which is a viral evolution during the pandemic. And these are all focused around antibodies and what antibodies do. So antibody therapy has been around for an awfully long time, including giving horse antisera to the toxins of diphtheria and tetanus. But despite the fact that there's more than 100 and now almost 120 years of experience in this, there are really not a lot of antibodies that are used in human therapeutically. And here's a slide showing all the different antibodies across the bottom that are used in human medicine, but only a few of them, the ones in red, are in infectious diseases. And that's a little bit surprising, actually, because of course, antibodies are fundamental to the control of infectious diseases. And so being able to harness that particular effectiveness, the effectiveness of antibodies to help meet unmet medical needs, it's an important goal. I think that there's now a strong basis that this is going to be a big deal going forward, and that we'll see more and more of that. And an example is this drug, Synagis, which is for respiratory syncytial virus. This is a drug, an antibody that you could administer to babies who are at special risk of RSV-induced disease, and it really works. Now there are a number of similar antibodies that have been tested. So there's a lot of potential here, but the potential hasn't been met. So I'm just going to give you an anecdote about an antibody that was isolated by some scientists at Vir, which is an Ebola antibody called MAB114. And MAB114 has been now tested against Ebola, and it has some pretty surprising effects. So the antibody is this yellow and brown crystal structure shown here, kind of nestled in between the spaces of the Ebola glycoprotein. The glycoprotein is what Ebola uses to enter cells and then to fuse with the endosome and release its genetic material into the cell. And this antibody interacts with a very specific part of this structure, and it has a profound effect on Ebola infection. And not all antibodies do this, but this antibody actually can be given to, now we know a human, but originally a monkey, with severe Ebola disease, and it can reverse the disease. So it can actually be a therapeutic. It can treat an established viral infection. And so this is really a pivotal paper for antibodies in the field. In December of 2019, there were 4 different drugs tested during the huge and unfortunate Ebola outbreak in Africa. And the drugs were a small molecule, remdesivir, which you learned about that type of drug last week, I guess, and 3 antibody cocktails. They're not all cocktails. Actually, one of them is a single antibody. And one of them is a combination of 3 antibodies, and the other one called Regeneron EB3 or REGN EB3. That's a combination of 3 antibodies. So this was administered to people with Ebola, and the characteristics are down here. It's a single antibody given 50 milligrams per kilogram in 1 infusion. So you just dilute the antibody, infuse it, and it has a very substantial effect on Ebola virus-induced death. Regeneron, their cocktail worked equally well. And so 2 things you learn from this. 1, you can treat a bad infection with an antibody. And 2, you can have one antibody that does the job, or you can have a combination of antibodies that do the job. And there's a lot of work going on and a lot of people saying you have to have more than 1 antibody, or maybe 1 antibody will work. The bottom line is the question's already answered. Single antibodies can work, multiple antibodies in combination can work. The key thing is you got to have the right antibody, okay? Not all antibodies have the kind of remarkable properties of this single antibody MAB114 or this combination of 3 antibodies, EB3. So with that as a little bit of background, I want to talk about the SARS coronavirus epidemic. Now, if you're going to use antibodies in therapy and prophylaxis of the disease, it's important to know something about the human antibody response. And so there's going to be 3 major kind of broad subjects in this session. 1 is the human antibody response, 2 is specific antibodies and how they work, and 3 is the evolution of the virus that is somewhat threatening from my point of view. So, you know, industry has this reputation that maybe they don't publish stuff, and I put this up here for 2 reasons. 1, I'm proud of this, and so give me a break on showing something that I'm just proud of. But 2, I just want to tell people that companies can actually really participate in the intellectual enterprise. So these are the papers that have come out of my colleagues at Vir, some with my participation and enjoyment of them. You can see they've been published in a wide range of journals. We talked about antibody mechanisms and antibody enhancement of disease in a review early on. We showed that antibodies can be made into vaccines, and I'll show some data about that. Then the antibody response to coronavirus, that's the next section. And then we found some pretty remarkable antibodies that we are taking into the clinic. They haven't been proven to work yet, but they're in advanced clinical testing now. And then additional mechanisms of antibody action. And this is a paper which is out online, but is in revision at a journal showing the first, what we believe is the first immune evasion mutation in the virus. So the SARS coronavirus, from the antibody point of view, there's really one protein that's important. It's the spike protein. And this is a structure shown as a ribbon, a space-filling ribbon diagram. This is a trimer. And so there are 3 proteins made together as a trimer that make this spike protein. You're seeing it from the side, okay? And now you're seeing it from above. And it kind of has this central circular area made of the 3 trimers and the 3 colors. And then this flower petal-like structure that kind of sticks out. I'm not going to talk a lot about that, but there's interesting antibodies to this so-called domain A as well. Now, one of the things about the coronaviruses is that they have learned to hide the part of their structure, their spike protein. This is the structure that interacts with the receptor on the cell, which is ACE2. They hide the part of their spike protein that interacts with the receptor as, I think, an immune evasion approach. And so sometimes these 3 domains called receptor binding domain, this top thing, sometimes they're down facing, and then sometimes one of them pops up, or sometimes 2. When they pop up, that reveals a special part of the spike protein called the receptor binding motif. And that part interacts with the receptor. So basically, this form is inert, and this form is active and fosters infection. So throughout the whole talk, we're going to be focusing on different parts of the spike, and there's the receptor binding domain. That's this kind of circular thing here, and the receptor binding motif. And that's the part that's revealed when this piece pops up and then can interact with the receptor. So this is the tool that the enemy uses to invade cells. And we're going to try to counter that. Now, how do people make immune responses to this? So one thing that we've done is to take samples from a large number of patients with different forms of the disease and do what's called high resolution serology. So what we did is we isolated a broad number of monoclonal antibodies. These are single antibodies that we express from their DNA sequence. And then we can study the properties of each antibody molecule individually. And each of these colored things here, that's an antibody in a cryo-EM or crystal structure interacting with the top of this spike protein, which is the gray thing here. Now, you can see they bind in different places. So they're different domains, and they each recognize a different place. And we picked them because they all recognize different places. Now you can do a pretty simple experiment, and that is that you can purify the spike protein, add the antibody, and the antibody binds to the spike protein with certain chemical characteristics. You can measure quantitatively that binding. And then you can add the serum from all these different people and ask, what kind of immune response do they have to each piece of the spike protein? And the part that an antibody recognizes is called the epitope. And so this is now going to be epitope-specific serology. We're going to ask, what do people actually see when the antibody responds to this spike protein? And so the spike protein can be broken up. This is the whole spike protein, the RBD, receptor binding domain, domain A, that flower-like thing. This is the stalk. This is a control in the nucleoprotein and no antigen. And these are different forms of antibody, IgG, IgA, IgM. And you can see that people with different characteristics, hospitalized, symptomatic, asymptomatic, or controls, have a lot of antibody. But you can also see this is a log scale. So these are 10-fold variations. Look at the huge variation in the amount of antibody. It's thousands to tens of thousands of fold. So the first message is there's an enormous variation in the human antibody response to this pathogen. And that's shown here. This is the total antibody binding to the spike on one axis, and this is binding to the nucleoprotein. And you can see that people have this huge variation. All right. We don't know how much is needed to protect from reinfection. These are all people who recovered and who didn't die. If we look at how sick they were, you can see that the severely infected people make much more antibody in general than the asymptomatic individuals. So that means the amount of exposure to the virus has a profound effect on the amount of antibody that people make. So huge variations, and the variation is related to the clinical syndrome that people have. How long does the antibody last? Well, we studied over 600 people in this study, and we found that the half-life of the antibody is about 49 days. So at the end of 49 days, you have half as much antibody as you did before. 49 days later, you have half of that again. By the end of a year, it's down more than 90%. And so some people, this is a graph showing the variation from the first time the serum was collected, which was within a few weeks of infection. And most people drop their titers quite a lot. A few people make these really nice responses where the antibody goes up, but that's not everybody. So the antibody is decaying fairly rapidly. This is potentially important because the common cold coronaviruses, which are related to SARS-2, immunity to them only lasts a year or two. And so we wonder whether this decay may have significance. We don't know that it does. And we're only talking about antibodies today. T cells are not measured here, and T cells may be protective independent of the antibody going down. So this doesn't say that people are not immune, but it does show that the antibody response wanes with a 49-day half-life. So a lot of variation, wanes fairly quickly. What is this antibody response? So now we're going to go to the epitopes. So each of these colored guys is an antibody. The black outline here is the RBM. This is the part that interacts with the receptor. And you can see that these antibodies overlap or don't overlap with that domain. And now this is hospitalized people, symptomatic or asymptomatic people, and how many make a response to that given epitope. So you can look here at this color, that's this antibody, that's this epitope, and about 98% of hospitalized people make a response. That's what we call immunodominant, okay? 100% of people make a response to this antibody, the orange one, immunodominant. But now you look at these other epitopes, and there are many fewer people respond. Those are not immunodominant epitopes. And then you look hospitalized, symptomatic, and asymptomatic. The asymptomatic people make a pretty poor antibody response. Now, why do I focus on this immunodominance thing? So the key question is that good or bad? And so first of all, it's good because that epitope, those epitopes, those immunodominant epitopes, that's the part of the spike that interacts with the receptor. But I'm going to make an argument that this is maybe bad. And that's coming up here in immune evasion. So viruses evolve in response to antibodies. So the fact that they have displayed something that's so immunodominant doesn't mean that we shouldn't trust the virus to do what's to its own good. So what if you could, if you were the virus, speaking teleologically now, what if you could mutate the heck out of that domain and still have it work? Now it's immunodominant, but you can mutate it and you can evade those antibodies. That's what influenza does. That's why people get influenza year over year. It has a part of the protein that elicits whopping antibody responses that protect you in a given year, and then the virus mutates and evolves away from those antibodies the next year. So immunodominance is good if you hit the right part, and immunodominance can have a second face, a Janus-like negative face, if the virus can mutate that part of its protein. So we'll come back to that. This is if you total up the antibody responses to all the epitopes of a whole group of individuals. And now you can see even more clearly, some people just make a very poor antibody response. Now, me personally, this worries me, but it also, I hope, that it means that they have good T cells and that they don't need these antibodies. But it's pretty clearly true that some people just don't make a good antibody response. And that's more frequent in symptomatic outpatients and asymptomatic people than hospitalized people. This is a wide variation in antibody responses. And the therapeutic and prophylactic use of monoclonals, the object is to give everybody a high level of a protective antibody so that you don't have to depend on them to make their own response. You're going to passively transfer, you're going to give them what they need. Now, how about the function of these antibodies? Now, this is this upward facing part of the spike protein right here. And this is the ACE2 interact, that's the receptor interacting. So you can measure the amount of blockade of binding of ACE2 to this receptor binding motif that's revealed when this thing pops up. And you can see that if you have a lot of antibody, this is total antibody on this axis, you have a good chance of making antibody that blocks this interaction. But there's really a lot of people who make antibody responses that don't have this property. And this is actually neutralization. So neutralization is the blockade of infection of new cells in vitro. And you can see some people make a good neutralizing antibody response, more common in hospitalized people in the red, but there are a lot of people who don't. So again, huge human variation in the antibody response. So what matters about antibodies other than the fact that they block the receptor? So this I'm going to spend a few minutes on. This is designed to help conceptualize how to think about whether an antibody is good, bad, how it works. And I've divided it up into 5 domains here. First, the antibody has to interact with different forms of the virus and the viral spike. For instance, the closed conformation or the popped up receptor binding motif conformation. There's neutralization, the ability of an antibody to block infection. There's things called effector functions. So the antibody is not just neutralizing. Antibodies are tools that the immune system uses to recognize a pathogen, but they also recruit the immune system to do its job. So neutralization is because the antibody binds to a critical part of the virus. An effector function is that that antibody is now hanging off the virus or the virus infected cell, and it's calling the immune system to do its job. Now, if you give an antibody, it may select resistance variants that make the antibody not bind. So this is the spike protein in blue and one of the clinical antibodies that I'll show you a little bit about in orange binding onto the spike. So the antibody may put selective pressure on the virus, and the virus may respond by mutating the spike so the antibody doesn't bind anymore. That's called a selected variant. And it could be, though, that the virus is mutating during the pandemic. Those are natural variants. So all of these domains have good news and bad news. It's good if you bind all the forms of the virus. It's bad if the dynamic structure or other changes make your antibody not bind. It's good if it neutralizes, but it's really, neutralization only deals with the free virus. It doesn't kill infected cells. And it could be that the virus is actually, I'm going to speak teleologically here, that it wants you to recognize this because it can mutate it. And so it can mutate the neutralizing epitopes and still survive as a virus. So that would be bad. It's good if effector function kills infected cells, but it's bad if the effector function causes inflammation and harms the tissue. It's bad if the antibody easily selects variants. It's good if the epitope that it binds to has a low mutation rate. It's bad if there are natural variants circulating in the population that the antibody doesn't bind. And it's good if the epitope has a low mutation rate. Note that for variants, low mutation rate of the epitope that the antibody binds, that's a good thing. So when we started out, we're very aware of literature showing that single neutralizing antibodies or even combinations of antibodies that target SARS-CoV, this is not SARS-CoV-2, the COVID-19 virus, but SARS-CoV from the original SARS epidemic, they show plasticity. And when combined, they have very rapid selection of mutants. And so that told us that these viruses can mutate in response to neutralizing antibodies. So the principles that we took were to find a conserved epitope, to use potency as best we could, to select antibodies that might be useful in combinations, and to fully map those neutralizing epitopes and understand how they could vary. So that's the approach. And we found an antibody that I'll show you more details on, which is called S309, published early on in the pandemic, and it recognizes a highly conserved epitope. And it neutralizes, and it actually neutralizes both SARS, the original virus, and SARS-2. So whatever it's binding is highly conserved across all of what are these sarbecoviruses. So the sarbecoviruses are 3 clades within the coronaviruses. And within the sarbecoviruses, you find SARS-1 or SARS, the original, and SARS-CoV-2. So this antibody recognized an epitope shared across those viruses. When we looked, this antibody called 309 was very potently binding to the spike protein on infected cells. And then we looked to see if it neutralized, and it was potently neutralizing, and that was good. And we looked to see whether it neutralized what are called pseudoviruses. So pseudoviruses are not the live thing. The live thing is BSL-3, and so shouldn't be worked on outside of high containment environments. But you can take that spike protein and put it on another virus backbone, and you can actually have the infection by that second pseudovirus, dependent on the spike protein. And it turned out that this antibody neutralized spike proteins from bats and SARS-1 and SARS-2. So it was very broad. And a very important thing is the concept of avidity. So antibodies can bind tightly but fall off quickly. Other antibodies bind tightly and don't fall off at all or minimally. And this antibody has an extremely high avidity measured by the dissociation constant here of picomolar levels. So this antibody, once it binds, it doesn't really fall off. That'll come back later. When we solved the structure of it, we found that the antibody, shown here in blue and red, looking down at the spike, bound to the spike in a part that doesn't block the ACE2 interaction. It actually neutralizes the virus, but it binds to this conserved place on the surface of the spike. So it didn't bind the receptor binding motif, which many neutralizing antibodies bind, but it did neutralize. Now, this is the phylogenetic structure of the coronaviridae with the MERS virus. That's the one that is harbored in camels in the Saudi Arabian Peninsula, and then SARS-1 and SARS-CoV-2, the cause of COVID. There are lots of viruses in this. And so let's look at the epitope. So this is now the amino acid sequence of where that antibody S309 binds. And you see it binds the top amino acid sequence is SARS, and the bottom amino acid sequence is SARS-CoV-2. And you can immediately look and see when there's a dot, that means it's the same amino acid in both viruses. This brown epitope, which is where 309 binds, highly conserved. Let's just look at the blue area. This is the receptor binding motif. This is the amino acid sequence of SARS. This is SARS-2. You don't have to be a rocket scientist to look at these sequences and say, wow, there's a lot of amino acid variation there. And the key point about these viruses is that unfortunately, despite that amount of variation, they still both use the same receptor ACE2, and their spike protein binds ACE2 with very high affinity. So that means that SARS-1 and SARS-2 solve the same biological problem, binding to ACE2 with very different amino acid sequences. So this domain is less conserved between the viruses than this domain or this epitope. And this is just shown across members of all 3 clades. This is the actual amino acids that are contacted by that antibody when it binds. And you can see that across a very large number of viruses, pangolin viruses, bat viruses, this epitope is pretty conserved. So that's good. We like that it's a conserved epitope. What about these effector functions? So these are some cartoons that show this concept. So this is an infected cell releasing viruses, these red people here. And these are the antibodies binding, and they're neutralizing the virus and preventing the infection of a new cell. So this is neutralization. Neutralization is good. Neutralizing antibodies that bind to a highly variable part of the spike protein may be less good over time, although they can be extremely potent, and many of them are. But there's the added problem that you can get rid of these viruses outside and save the new cells. But unless you get rid of the virus factories, you keep producing more virus. So effector functions. This is the role of antibodies in recruiting the immune system. The antibody can bind to receptors on macrophages and NK cells through this portion of the antibody schematized here. It's called the FC portion of the antibody. And the antibodies can neutralize the virus, but they can also bind to the viral spike protein on the surface of cells, and it is expressed on the surface of cells. So they bind to the infected cell and then call in a macrophage to phagocytose, eat and kill the cell, or a natural killer cell to come in and kill the cell. So an antibody that can neutralize and have effector function may be more potent than an antibody that neutralizes only. So that's why we think effector functions are important. And this is shown in a very nice paper, which you can look up online, Schaffer et al. And JXMed. They looked at the efficacy of a series of monoclonal antibodies against SARS-CoV-2 in a mouse model. It happens to be a very sophisticated mouse model that is highly representative. And these are all different antibodies. And you can see the amount of virus in the lung. So these antibodies don't work very well. There's a lot of virus in the lung. This antibody works very well, very little virus in the lung. And this is neutralization potency. If there's a low number, it's very potent at neutralizing. If it's a high number, it's not very potent. And so what you can see here is that the best neutralizing antibody is substantially less effective than this antibody, which doesn't neutralize very well. But this antibody has those effector functions that enable it to recruit additional parts of the immune system. So antibodies are dual function. They neutralize and they have effector function. You can actually prove this by taking those antibodies and putting a specific mutation in that FC part of the antibody and deleting effector functions. And then this antibody, potently neutralizing antibody, when you mutate effector function, makes no difference. It works by neutralization. But this antibody, that very potent one that really controls the viral titer, if you mutate it, it loses potency. So when you mutate effector function, it loses potency, as does this antibody. So antibodies they're dual function. They can neutralize, and that can be good, and they can have effector function, and that can be good. The antibody 309 happens because of the orientation of how it sits on that spike protein. It has very potent effector function. It has so-called antibody-dependent cell-mediated cytotoxicity. That's those NK cells killing infected cells, or in this case, cells expressing the spike protein. And this is phagocytosis of cells expressing the spike protein. You'll note that a lot of other antibodies don't do this. And in fact, many of the most potently neutralizing antibodies don't have these effector functions. And we think we have some idea about that, but actually we don't fully understand that dichotomy. But this one does both neutralizing and has effector function. So if effector function is important, can we beat nature? Can we do better than nature by engineering that part of the antibody to enhance effector functions? And this is a concept which has been pioneered by a scientist named Jeffrey Ravitch and his co-workers at Rockefeller University for several decades. And I'm going to show you some pretty striking data that was published just recently in Nature with Jeff Ravitch as the senior author and some Vir scientists as well, where a new concept was tested about, can you make an antibody work better? So these mutations that I'm going to show you don't change neutralization. Neutralization is exactly the same because the part of the antibody that sticks up, binds to the virus and neutralizes, no difference. But that FC part that interacts with the immune system, that's being mutated and changed. So why would we do that? Well, it turns out that that FC part of the antibody binds to a whole series of Fc gamma receptors. These are receptors on the surface of the cell, and all of them bind the Fc part of antibodies. And what you can see right off the bat is that there's a very complicated process here. The body has evolved multiple of these receptors. They must have a purpose. In a non-Nietzschean universe, if they're genes and they're conserved across species, they probably do something, okay? And it turns out that these different Fc gamma receptors that interact with antibodies have different effects. This one affects dendritic cells, come back to that. These two are effector functions, cytotoxicity, for example. And this one, Fc gamma R2b, actually inhibits immune responses. So it's a down modulator. It's a brake on antibody-mediated effector functions. So what if you mutated an antibody so it bound better to activating receptors and bound worse or not at all to an inhibitory receptor? And the concept here is that they're antigen-presenting cells in the body called dendritic cells, and their job is to present antigen to induce T cells. So would it be possible to make an antibody that neutralized a virus, but somehow manipulated this complex system to turn on antigen-presenting dendritic cells and make the antibody become a vaccine so that it vaccinates somebody against their own infection? It's a pretty cool idea. And it's been something that drove Jeff Ravitch's science for a long time. So this is an influenza experiment, and this is done in mice, but the mice have been engineered, so they only have human Fc gamma receptors. All the mouse receptors are gone, replaced with human receptors, which, as you might imagine, took a while because there are a lot of receptors. And we're just going to give an antibody to these mice, different doses of the antibody, and give them influenza virus. And you can see that if you give a high dose of antibodies, the mice don't lose weight. But as you lower the dose of antibodies, the mice lose weight and are sacrificed. Now we're going to put this mutation called XX2. XX2, it's now been published. It's GALA-E. You can look up the amino acid changes in the paper that's referenced here. And now look, the same dose where all the animals did poorly, they're fine. All the animals did poorly. Some of them do poorly, but the others recover. And so this antibody is much more potent at protecting these animals from a respiratory virus. In fact, across all these experiments, it's about 5-fold more potent. And that's a huge difference. That means that you could conceivably give 1/5 the amount of antibody and still have an effect. Now, this is a prophylaxis experiment. In other words, the antibody was given before infection. How does it work? So this is some pretty remarkable immunology. First of all, is it true that the antibody activates dendritic cells? And the answer is yes. These are dendritic cells taken out of infected animals, and when you treat them with the antibody, then dendritic cells are more active, shown by cell surface markers. How about T cells? The number of activated CD4 T cell is actually higher, meeting expectations. And does it matter? Does all this immune induction, this vaccinal effect matter? And so that's shown over here. Let me take you through this. This is the wild type antibody. So this is a protective antibody, and it's dosed at a level that protects half of the animals from dying. That's the black line here, okay? Now, if you give the vaccinal antibody in the same dose, all the animals live. So this difference from black line to solid red line, that's the increased potency brought on by engineering the Fc part of the antibody. So now let's ask, do these T cells matter? And so what we're going to do here is we're going to take the animals which receive the vaccinal antibody or the control antibody, and we're going to deplete the CD8 T cells. And look what happened compared to the red line to the dotted red line. This is the vaccinal antibody. This is the vaccinal antibody, and the animals have had the CD8 T cells, the killer T cells, deleted. And now the antibody behaves just exactly like its parent, just neutralizing antibody. So this shows that you can make an antibody that elicits T cells that have potent effects. And perhaps even more remarkably, it's been shown both clinically in humans and also in animal models that it's very hard to treat influenza with an antibody. And so the blue lines here are the results of giving multiple different doses of this potently neutralizing antibody to animals that have already been infected for 3 days and they've started to get sick, and the antibody doesn't do anything. But the vaccinal antibody saves them. So you can actually treat an infection with this engineered antibody in animals. This is reminiscent of that data that I told you about with Ebola. And that is that there are certain antibodies that you could give and actually reverse or treat severe virus infection. And so the conclusion that we draw from this is we love neutralization, blocking new infection, good idea. Effector functions are important, but there's an opportunity to improve on nature here. And just if you think about it, why is that? And the answer, I think, I'm going to just purely speculate here. So it turns out that we make antibodies to our own self-antigens all the time. I think it would be pretty poor idea for our own antibodies to vaccinate ourselves against our own antigens. So for instance, if you have thyroid trauma in a car accident, you can make antibodies to your thyroid proteins. If you had a vaccinal antibody circulating, you could become autoimmune to your thyroid. That's a bad plan. So evolutionarily, it does not make sense to have antibodies be vaccinal. But in this case, the antigen is outside the body. So we can improve on the response by doing some manipulations that allow us to unleash the natural immune system in a setting where we're not worried about autoimmunity because the antibody is specific for a pathogen. So that's the idea of engineering antibodies to make them better prophylactics, better therapeutics. So in the last 10 or 12 minutes, I'm going to present some data that I find to be less optimistic, okay? And this has to do with whether the enemy, SARS-CoV-2, is up to the game of mutating to get away from antibodies, all right? Now, there's a lot of stuff that you read in the press about this virus mutating slowly. I would like to debunk that. I think it is this is my personal point of view, I think it is an incorrect assessment of the science that it's written that this virus mutates slowly. And I want to talk about that. And I want to show you an example of something the virus has already done to evade antibody-based immunity. So remember that there's this wide variation in coronaviruses within the sarbecovirus. And some of the epitopes are conserved between SARS and SARS-CoV-2 and a whole bunch of bat and pangolin and other zoonotic or animal viruses. Remember that the conserved epitopes can be recognized by antibodies, but antibodies that bind to this part, they can be very potently neutralizing. They can have some effector functions. Many of them do not. But this epitope seems, just from looking at this, to be mutable and still function. So what controls the rate of variation of a virus in a pandemic? The first thing is what you read about in the press, and that is the viral polymerase. Certain viral polymerases make a lot of mistakes. The HIV polymerase makes a lot of mistakes, okay? DNA virus polymerases, and thankfully our own DNA polymerases that we use to replicate DNA, they are not error prone. They're pretty fastidious or processive in their functionality. They don't put the wrong base pair in frequently, but many viral polymerases put the wrong base pair in frequently. In fact, it's been shown in polio that if you make the polymerase more processive, more fastidious, it interferes with the capacity of polio to cause disease. So the virus is actually intentionally making mutations frequently. And that's just to try out evolutionary space. It turns out that the coronavirus polymerases are not as good as the DNA virus polymerases or R polymerases, not as bad as the HIV or polio polymerases, but they do make mistakes. But that's not what controls mutation in epidemic. The virus has to be fit to transmit and replicate. If for disease-causing viruses, it needs to be virulent. It needs to, these mutations are under selective pressure. So the frequency of mutation is irrelevant except in the presence of pressure. And antibodies, B cells and T cells provide pressure. So far in our pandemic, there's no substantial herd immunity. So the virus is mutating in millions of people around the world, as yet not under immune pressure. So there's no significant immune pressure on a species or quasi-species of viruses until herd immunity emerges. Because until you have herd immunity, the virus can easily spread to a non-immune host. So whether there's an immune response doesn't matter to the virus in a certain sense. Now, of course, the number of mutants is not determined solely by the processivity of the viral polymerase, but by the sheer number of infected people. So right now, we as a species of humans are unfortunately participating in one of the largest viral evolution experiments ever done, because we've released a virus that no one is immune to. Maybe a few people are immune to. Let's set that aside for a moment. Very few people are immune to. And now it's infecting millions of people and varying around the world in all those people, trying out new amino acid sequences to see how things go. And then so selection occurs by herd immunity, but it could also be driven by vaccination or therapeutic antibodies. Remember, if the antibody blocks the virus and the virus can mutate away from the antibody, then the virus, as long as it's fit and can transmit, it's going to mutate. You can trust the virus to do what is in its best interests for transmission and spread. So this is an image of the receptor binding domain, and the blue is the receptor binding motif, that part that I showed you that many neutralizing antibodies bind to. The S309 binds over here. And this is just across the more than 150,000 sequences, the rate of variation in different parts of the spike. And it turns out that this receptor binding motif varies faster than any other part of the spike. Okay, this is highly reminiscent of the head of the flu virus hemagglutinin. The hemagglutinin elicits very potently neutralizing antibodies, but the virus can mutate it very quickly and it still functions just fine in influenza infection. It's almost as if the flu has made a decoy. It's put something out that the immune system recognizes, but it can mutate. It's plastic. It's a protein that exhibits functional plasticity. That is, that it can function with many different amino acid sequences. So that's a concept that comes from influenza. May relate here. So now I'm going to tell you about a variant that we think is the first well-characterized immune evasion variant of SARS-CoV-2. So firstly, this is the crystal structure of the ACE2 in the orange and SARS-CoV-2 below. And much of the energy comes from two salt bridges. Okay, SARS, the original SARS, uses this, this salt bridge, and SARS-2 uses this salt bridge. What would happen if the virus evolved to use both salt bridges? Well, it turns out that that has happened. N439K takes this mutation, which was N, and mutates it to K, which creates a charge that interacts with this charged residue on ACE2. And so now the N439K SARS-CoV-2 isolate uses both and binds even better to the receptor than viruses that use only one salt bridge. And what happened is that this was detected by our colleagues. You can look on the paper on bioRxives. It was detected in Scotland, and then by social distancing and mask wearing. The function of a wise society at protecting each other is doing those sorts of things. And it actually cleared out of Scotland, but unfortunately it re-evolved, and now it is in all of these countries, and it's increasing in frequency worldwide. It's now the second most common variant in the databases. And so this now took a single salt bridge and a single salt bridge and made it into two salt bridges. And this is shown biochemically here by showing that you can measure the affinity of the interaction of this domain to ACE2, and it has a lower affinity if it has no salt bridges. If it has one salt bridge, it has certain affinity, and if it has two, it has a higher affinity. So this virus receptor interaction is stronger with this mutation. Now, this is a charge mutation. It's a charge switch, which is a pretty big amino acid change. It turns out that this virus is virulent. And so if you look at people who had the N or the K residue at 439, and you look at the proportion of them who died, had severe need for oxygen, low need for oxygen, they're identical. So this is a virulent virus. It's fit. This is the replication of the virus in the nasal passages of people. This is beautiful work done by a group in Scotland that contributed to this paper. Their scientists did a lot of the work on the structural biology. It was a wonderful collaboration between this academic group and a network of scientists in Europe and our scientists. Bottom line is this virus grows fine. This is the actual growth of the virus. It may grow a little quicker, but it's certainly not compromised. And this is a classic test of fitness where we competed the viruses against each other, and the virus with the new mutation competes effectively against its wild type parent. So this is a fit virus. What about the immune system? So we took sera from that big group of people, and we showed that there's a significant number of people who drop their titers below what we believe to be significant against this. So the single amino acid change changes the serologic response, the total serologic response, to just under 10% of people. And then we took 144 different monoclonal antibodies and tested them, and we found a bunch of monoclonal antibodies that lose their binding. So these are all RBM antibodies. And then we did the following experiment. So we took clinical antibodies. So these are antibodies in the clinic already. S309, I've shown you about, that's a Vir antibody. LyCoV555, that's the antibody that Lilly first put in. It's now part of a combination. There's nice data that's been released showing that this antibody works on prophylaxis. Regeneron took forward a combination of two antibodies, which also works. And this is a control antibody. And these are binding curves. You can see binding to whether it's the first salt bridge, the second salt bridge, or both salt bridges are mutated. These antibodies bind fine. Okay, so these clinical antibodies don't care whether it's N439K, but this antibody does. It's a clinical stage antibody, and this is neutralization. It loses in the range of 10 or so fold or more neutralization capacity. So this mutation evades a clinical monoclonal antibody. And that's shown here in a rather complicated figure. It's my last one. And this just shows the fold change in neutralization when you make these mutations. And you can see a lot of antibodies just don't care. Okay? But certain antibodies lose their ability to neutralize. Their neutralization titers go way up when you mutate one or both of these. And here's one of the Regeneron antibodies. And this is the Lilly antibody, and this is the other clinical antibody from Vir. So what we've shown here is that the virus definitely can evade antibody immunity. There's a fit virus circulating around the world, increasing so far in frequency. It's virulent. It grows normally in people and in culture, and it evades the immune system. In this case, it evades certain monoclonal antibodies and even a polyclonal antibody response from some individuals. So I'm not proposing that this is going to be the future of what's going on here. I don't think anyone knows what's going to happen with this virus, but the virus clearly has the capacity to evolve in a way that can evade at least some antibodies. I don't have a crystal ball to tell you that this kind of property will influence vaccines or will influence antibodies. I do think that it is a good idea for us as a species and different countries and different scientific institutions to track the evolution and immune evasion properties of this virus very closely. We all need vaccines, antibodies, therapeutics to work. And if the virus can mutate already before there's immune pressure, I think we can at least be aware and prepared for when it happens, if it happens in the real world. So that's my last slide. I've shown you that neutralization, effector function, and variants are all key elements of how you think about antibodies. I've shown that maybe we can improve on nature a little bit by engineering antibodies, and that's kind of an interesting idea. Science is not done in the pandemic except through broad collaborations, and acknowledged here. We have collaborations with a very fine scientist at Washington University School of Medicine, Michael Diamond, who's contributed to these studies, a wonderful scientist, David Wiesler, at the University of Washington, and who does a lot of beautiful structural work. We have collaborations with KU Leuven for animal experimentations. And of course, this kind of science is only done by very large groups of people who are extremely dedicated, working long hours in a BSL-3 at personal risk in order to try to help deal with the pandemic. And with that, I'll stop and take any questions that people might have.
Thank you very much, Skip. We have a whole bunch of questions. I'll try and summarize the key ones. What's the half-life of antibodies administered to humans?
Yeah, that's a really good question. I didn't go into that. So it varies in the range between 2 and 4 weeks. Think about it in 3 weeks or so. And so that's, it varies by the sequence of the antibody. There are mutations I didn't go into, which are in some of these clinical antibodies, including the one that we're taking forward, which specifically lengthen the half-life. So another one is in the antibody that's been taken forward, as we understand it, by AstraZeneca. And these extend the half-life so that a single dose lasts at therapeutic or prophylactic levels for a longer period of time. So there's antibody engineering to extend that natural half-life.
Are you thinking that prophylactic antibody administration may be feasible on a large scale, or is it you thinking this is more useful as a therapy following infection?
I believe that antibody functions both therapeutically and prophylactically, and that it is practical. I think you have to solve problems like it has to be administered easily. It has to be formulated so you can give enough antibody to be prophylactic for a long period of time. But I do believe that antibodies can be used for both. It has been proven that that is true. The Ebola antibody used for therapy and the RSV antibody that I introduced, Synagis, in the very beginning, that's a prophylactic antibody. That's administered and prevents vulnerable babies from getting severe respiratory disease from RSV. So I think both uses are on the table, and both uses are being tested by multiple companies today. So we're going to learn a lot over the next months.
And if you have a virus that's evolving and you anticipate it might evolve to escape your favorite antibody. Can you imagine a scenario where in subsequent years there's a change in that cocktail, that either the single antibody or a mixture of antibodies? And are there issues associated with that? Do people develop antibodies against the antibodies? Are there dosing issues?
So there are two questions in there. One is maybe antibodies will need to be changed over time. I don't think anybody knows that, but I think that some of the clinical antibodies that are out there now have already been proved and published by their originators to rapidly select escape mutants. And then they're used in combination. So the Regeneron antibodies are a combination of two antibodies that are that each individually have been shown in their own work can select these variants. And so that's a combination. And that's a good idea. When you have antibodies that select variants rapidly, then having a combination is good. I think the jury is out. We believe that having an antibody to a conserved epitope that's shared across all the sarbecoviruses may give the opportunity to treat or prevent virus infection for a long time during the pandemic. But, you know, you always have to assume the worst when you're dealing with a pandemic. So I think everyone recognizes that we need to be prepared for that variation should it arise. Same thing is true for vaccines. The vaccines may work or they may need to be altered in the future. I don't think people know the answer to that. The only way to responsibly take on a pandemic is to assume that the virus will do its worst and be prepared for it as a culture and as a species.
And on that note, Skip, I have to thank you very much for your presentation today. It's been wonderful to have us.
I hope some of the people in the audience take on the pandemics of the future and do great science, great development, and put in place policies through which the governments and other institutions in our cultures can address future challenges. That's the job of the people listening out there. Do a better job than we've done.
Thanks so much, Skip.